Did you know that you share 50% of your DNA with the banana? You might be more closely related to any fruit. This is because all living cells contain genetic material in the form of chemical DNA. Each of your cells contains an incredible 2 meters of DNA. It's possible to fix all that DNA in a tiny cell because DNA extremely narrow, only about 2 nanometers, and you around proteins. You can find out more about DNA and structure by watching this video. Do you want to see what DNA looks like? Well, it is possible. You just need to extract it from some cells. You can do this in the lab, even in your kitchen. The easiest way to do this is to use some fruit. In this video, we will go through the steps to show you how. First, you need to choose your fruit. The best fruit to use of those have plenty of DNA. Strawberry shows as a good choice, also kiwi fruit. Take 5 milliliters of washing up liquid. Take 50 milliliters of water. And 5 grams of salt. Then stir well. You then blend or smash the fruit in salt water. It broke apart the cells and was to start to break down the cell walls. So, tissues that the DNA is more likely to come together in the final stage. The next stage is, just pass the liquid through a sieve and collects the fruit juice. This will contain all the strawberry cells with the DNA inside the nucleus. To break apart the membrane surrounding the cell and release the DNA out of the cells, you add some detergents such as washing up liquid to the liquid. This ruptures the cell membranes. I think a pinch of protease enzyme will help to break down the proteins with DNA's wound tightly around. The DNA will uncoil now in the mixture as long strings. You can use pineapple juice, contact lens cleanup, or a pinch of meat tenderizer. These will contain protease enzymes. You can't see the DNA because it is dissolved in the mixture. To see it, you have to make it insoluble. And to do this, you slowly pour ice-cold ethanol into the mixture. The ethanol at some low temperature slows down the action of enzymes naturally present in the cell. That will break down the DNA. Ethanol is less dense than water, so it forms a layer on top of the fruit mixture. The DNA is insoluble in ethanol, so begin to precipitate out to the fruit mixture at the boundary between the fruit soup and ethanol. You will see cloudy strings of DNA that you can remove by winding around a wooden stick. Here is the DNA. You can then observe this DNA from a microscope or use with for experiments.